Today we start with the tools of DNA technology that we have left in the last presentation. Now the first tools for DNA technology was enzyme, and now the second tool for the DNA technology is exonuclease. So what is exonuclease? It helps in eliminating the nucleotide from the end of the nucleic acid molecule, that is from the 5' 3' end. Now, some of the examples of the exonuclease are exonuclease BAP131. It helps in utilizing the products like the DNA fragment with blunt end, which helps in shortening in both its end. Now, the second one is E. coli exonuclease 3. It is helped in used in 3' end modification, since it has the capability to remove the nucleotides from the 3' oxyl group of the DNA. And the last one is lambda exonuclease. It helps in utilizing to change 5' end of the DNA, as it eliminates the nucleotides from the 3' end of a linear or a single DNA molecule. Third tool for RDNA technology that is required is a restriction endonuclease. So what about this restriction endonuclease? How does it activate like a tool? Now it helps in the sugar phosphate support of a DNA strand. It helps in cutting the restriction endonuclease enzyme, which is isolated from a microorganism. They help in transmitting a host defense mechanism or a function from the cell. Now there are different types of restriction endonuclease. There are three basic types. The first is the class 1 endonuclease. It has a molecular weight of 3 lakh daltons. It is a non-identical subunit. It requires Mg2+, ATP and SAM as cofactor for its activity, whereas in class 2 endonuclease, that its molecular weight is 2 lakh to 1 lakh dalton, and its equal subunit, it has equal subunit. It needs Mg2+ as its cofactor, not ATP and SAM. The class 2 endonuclease does not require ATP and SAM as its factors for the activity, whereas the class 3 endonuclease has a molecular weight of 2 lakh daltons, which needs Mg2+ and ATP as its cofactor but not SAM. The most effective restriction endonuclease is type 2. Told that the most effective restriction enzyme or restriction endonuclease is type 2. Now we'll further see the mechanism how it is helping in enzyme digestion. Now here we can see we have a double-stranded DNA, and we choose or we tag a specific recognition site at the middle or at the end. So with the help of restriction enzyme, we are taking the help of restriction enzyme, that is homodimer. Now we further can see with the mechanism that is happening, it is forming into two different types. One is the non-specific binding is taking place. How is this non-specific binding taking place? With the help of linear diffusion. And in the other part we can see a specific binding is taking place with the help of coupling. Now this homodimer is with the help of this coupling method, it is getting attached to the DNA double-stranded DNA. With that we can see slowly with the mechanism, we can see at the last stage, last to last stage, that this homodimer are getting cleaved from the DNA strand, which is further releasing the homodimer at a site, and this DNA fragment or DNA double-stranded DNA is getting divided and a fragment of the double-stranded DNA is getting released. So the released product is the sum parts of the double-stranded DNA along with some fragments and the homodimer. The last tool for our DNA technology is the DNA polymerase. It has an immense importance in the whole rDNA technology. Now, how it is being used? It is an enzyme that helps in the synthesis of a DNA strand from an existing DNA or RNA template. Now, this DNA polymerase are of different types. The first is the clinofragment of DNA polymerase 1. It helps to create the producing ends of a double stranded by extension of a small strand. How can you do? Like we take a small strand, it helps in extension or extending the small fragment into a double strand. This is done by clinofragment of DNA polymerase 1. The second is tag DNA polymerase. It is very useful in PCR. PCR refers to polymerase chain reaction. We will further learn about PCR. There you will understand that what is the requirement of tag DNA polymerase. The third is reverse transcriptase. How it is helping? It is being used for RNA as a template which helps in synthesizing cDNA or which helps in creating cDNA libraries. The last three DNA polymerase are polynucleotide kinase enzyme. So what is its importance? It helps in reversing the influence to that of alkaline phosphate. The function is to add phosphate to 5' terminals or 5' end of a DNA molecule. It does nothing but it adds back the phosphate to 5' of a DNA. You know about the 5', 3'. At the 5' end, it adds the phosphate group. Now the last second is terminal deoxynucleotide transferase enzyme. It helps in enhancing the single-stranded sequence to a 3' terminus of a DNA molecule. One or more deoxynucleotide like DATP, DGTP, DIIP, DCTP. These are the types of deoxyribonucleotides which have been added onto 3' terminus of a blunt end of a DNA fragment. Now the last is alkaline phosphate enzyme. It helps in eliminating the phosphate group from the 5' M of a DNA molecule. In my previous slide or the previous presentation, I have told you about the cloning vector, the host or what is vector. I have told you in that presentation about a detailed study about it. Now I am giving you an example of a cloning vector. So here we have taken PUG19. PUG19 symbolize P. The P in the PUG19, you can see the name, symbolize plasmid, and UC symbolize as because PUG, this plasmid vector was being research done at University of California. So UC has been taken from that name, that is University of California, and this 19, that PUG 19, the part 19 part is symbolized that it has a high copy number plasmid. It can be copied at a high rate. Till date 19 number of copies has been yet researched. So from there the name has come up, that is PUG19, and it has a BP of around 2686 BP. And here now we come about the structure of the cloning vector is that it carries a resistance gene, that is ampicillin, AMP, resemblance, ampicillin resistance. The lag Z polymer, the lag Z symbolize that the N-terminal fragment of beta galactosidase gene of the lag Z E. coli. Now, this is the part that lag Z is the part where numerous restriction endonuclease, with the help of restriction in endonuclease, you can put many foreign gene insertion from this part, that is the lag Z part. And ORI symbolizes origin of replication. Now, this restriction endonucleases part is specifically for the lag Z, where many types of foreign gene insertion can be taken place with, and further you can do digestion. And this is all about this cloning vector of PUG C19. Knowing what is a cloning vector and how does it look like, its diagrammatic, its origin, amplification, everything, you have to know about its types. The types of cloning vector, there are different types of cloning vector, generally named as, there are 6 types. The first you can see from this table. This table shows everything like the name, the size, the cloning limit, the marker gene, and the example of the specific cloning vector. Now, the first cloning vector is an bacteriophage, which is of a size is of 4850 BP, and it has no cloning limit. There is no marker gene for bacteriophage, and the example is lambda genome. We generally use bacteriophage a lot very time because it is generated from E. coli. We take it as a reference because it has no cloning limit. You can clone as much as you want. It is unlimited. It has unlimited cloning, and you don't require any marker gene for this bacteriophage. Second comes plasmid. Plasmid of size is 4361 BP, and it has a cloning limit of 0.1 to 10 kb, and it has a marker gene that is ampicillin and tetracillin, and example is PBR322. We also use plasmid. We always use all these cloning vectors are being used in different types. We'll further see in the application. When we'll be learning about application, you all will be seeing the use of these different types of cloning vectors. Now, the third is cosmid. It has a size of around 79000 BP, and its cloning limit is 30 to 50 kb. It has no marker gene, and there is no example. No, sorry, the example is cos SI. And the fourth is human artificial chromosome. It has no size, okay, and there is no limit for cloning, and it has no marker gene, and there is no example for human artificial chromosome till date. And coming to the third, coming to the fifth, yeast artificial chromosome. Now, it has a size of 11400 BP, and its cloning limit is 100 to 1000 kb, and its marker gene is similar to that of yeast, and its example, there is no example for yeast artificial chromosome. The sixth is bacterial artificial chromosome, that is of size 11827 BP. Its cloning limit is 35 to 300 kp, and its marker gene is ampicillin and lactose metabolizing gene, and its example is PUV back. Okay, so PUVBBAC, or you can say PUV back. So these are all the types of cloning vector with reference to all its size, its limit, its marker gene, and its example. You have to learn and memorize this, very very important. Last part of the PPT, that is the linker and adapter. So you have to be well aware about the linker and adapter importance. It is generally it is like one of those of a cloning vector or host, like you can't do without them. Similarly, linker or adapter is very important. Now, coming to the linker mechanism, you can see when we add two linkers from two sides of the DNA strands. At the two ends, you can see at the two ends we are adding two linkers. Then the linkers are getting attached from further with the help of this DNA with the linker and with the help of restriction enzyme. We can see that there are two nicks, or there are two fragment nicks that are being formed. One is at the end 5 prime 3 prime, and another 3 prime 5 prime nicks are forming, which we when insert into a vector, you can see how this recombinant DNA looks like. With the vector, or within the vector, you can see how the restriction enzyme and how the DNA with the linkers are looking like. This is how the rDNA technology takes place with the presence of linker. Coming to the adapter mechanism, same way like the way we added both the linkers at the end, we are adding both the adapter to a foreign DNA. Okay, we are adding both the adapter to the ends of a foreign DNA. With the help of ligation, we can see similarly a 5 prime 3 5 prime 3 prime and a 3 prime 5 prime ends are being formed, that we are calling as the nicks. At the one end, it is forming a phosphorylation at 5 prime end is taking place, and which we further insert into a vector. And how the vector is looking like, you can see in the picture, it is forming a recombinant DNA with the help of the adapter. With the help of the adapter, a cohesive ends are being forming in place of a adapter. So these are the two examples or two mechanisms of adapter. If you have been asked what is the mechanism, you can write this. What is the difference? You will know the mechanism, that will help you to make it more simpler to understand. And further in my next presentation, I will be telling you about the rest of the parts, about the application and the polymerase chain.